Dear students, I am Bharat Kashyap, lecturer Biology at GSSS Majwar District Mandi. Today I want to teach you 8th chapter of Biology, Human Health and Diseases, and today's topic is AIDS and Cancer. Let us begin today's topic, Immunodeficiency Diseases, and in this we will study AIDS, acquired immunodeficiency syndrome in detail. Let us explain first immunodeficiency diseases. These are those where the defense mechanisms of the body become weak, which leads to repeated microbial infections. These may be of following types: primary immunodeficiency diseases. These diseases exist from birth. A person may be without B cells or T cells or both from birth. For example, SCID, severe combined immunodeficiency disease. It is most serious deficiency disease of children who lacks both B cells and T cells. These children are kept alive in germ-free isolation suits. Then second type of diseases are secondary immunodeficiency diseases. These immune deficiency diseases are acquired in lifetime by an individual. These may be due to some infections, metabolic disorders, drugs, etc. And for example, AIDS, the acquired immunodeficiency syndrome, which falls in the category of secondary immunodeficiency diseases. Let us study AIDS in detail. Let us study AIDS or acquired immunodeficiency syndrome in detail. Look at the diagrams here. These are the pictures of human immunodeficiency virus. AIDS was first reported in 1981 in USA, and in the last 40 years or so, it has spread all over the world, killing more than 25 million persons. AIDS is caused by the human immunodeficiency virus, HIV. It is a member of a group of viruses called retrovirus, which have an envelope enclosing the RNA genome as the hereditary material. It contains a reverse transcriptase enzyme which transcribes DNA from RNA. You can see the diagram here. This is the single strands of RNA, and this is the enzyme associated reverse transcriptase with each RNA strand, and it is surrounded by protein coat, and which is further surrounded by the lipid membrane. These are the glycoprotein present on it. So this is a HIV, and this is the diagram. You can see the internal structure in this diagram. So this is the structure of HIV and introduction of AIDS. Now let us discuss mechanism of action of HIV virus. After getting into the body of the person, the virus enters into macrophages. Let us discuss first what are macrophages. These are the cells of immune system and are formed from monocytes in response to an infection, and these cells are phagocytic and engulf infection causing viruses. Now RNA genome of the virus replicates to form viral DNA with the help of enzyme reverse transcriptase. This viral DNA gets incorporated into host cell's DNA and directs the infected cells to produce virus particles. The macrophages continue to produce virus and in this way acts like a HIV factory. Simultaneously, HIV enters into helper T lymphocytes to produce new progeny viruses. The progeny viruses released in the blood attack other helper T lymphocytes. This process is repeated and a progressive decrease in helper T lymphocytes occurs. Now let us discuss the mechanism of replication of virus with the help of diagram. Let us first see these are the monocytes which form macrophages. These are the WBC type monocytes and form macrophages. This is clearly visible in diagram here. Now let us see the picture. This is the retrovirus and this infect the normal cell. Now it enters into the cell. Then this viral DNA replicates by reverse transcriptase and form viral DNA. This viral DNA now incorporates into the genome of DNA of host cell and now infected cell it produces new viruses, this progeny viruses, and now this new viruses can infect the other cells. So this is the how viruses multiply in T cells, T lymphocyte cells, and decrease their number. Now let us discuss modes of transmission. AIDS is not a contagious disease. It does not spread through contact, carrying infected persons, shake hand, mosquito bite, etc. It may be transmitted through sexual contact with infected person, transfusion of contaminated blood and blood products, use of contaminated needles and syringes by intravenous drug abusers, contaminated blood transfusion, organ transplants, and from infected mother to her baby through parturition and breastfeeding. And now incubation period, the time between infection and appearance of AIDS symptoms vary from 6 months to 10 years. Now diagnosis. AIDS can be diagnosed by ELISA test. Its full form is enzyme-linked immunosorbent assay and Western blot test. ELISA is based on antigen antibody and enzyme substrate reactions. Now let us discuss various symptoms. HIV attacks helper T cells, reduce their number and causes severe cellular deficiency, so the persons become highly susceptible to bacteria and viruses. These clinically unhealthy persons are called opportunistics and suffer from opportunistic infections, also called OIs, like diseases of lungs, skin cancer, brain damage, hemorrhage, etc. You can look here the opportunistic infections, meningitis, there is liver and spleen enlargement, there is tuberculosis, diarrhea, pneumonia, and etc. There are many diseases also called opportunistic infections. Most serious form of AIDS is AIDS-related complex or ARC, which is characterized by swollen lymph nodes, fever, and loss of weight. Now, prophylaxis. As no vaccine yet available, so management of AIDS depends only on preventive measures. People, especially high-risk groups like college and university students, should be educated about AIDS. Blood tests must be done for donors of blood, organs, semen, and growth hormones. Disposable needles and syringes should be used. In sexual relations, one should be monogamous and avoid prostitution. Dentists should sterilize the equipments, avoid common blades at barbers' shops, avoid tattoos, ear and nose piercing from unqualified people. NACO, it is National AIDS Control Organization, and other NGOs are doing good job to educate people about AIDS. Every year, December 1st is recalled as World AIDS Day. Now, therapy. Although there is no specific therapy for HIV infections, yet some drugs like azithromycin (AZT) suppress AIDS virus. About 43 vaccines have been developed in USA, but none found successful yet. There are two ways suggested to prepare vaccines. These are antiretroviral therapy, that is ART. It helps in stabilization of AIDS symptoms of patient, but no cure from virus. Then immunostimulative therapy. It is to increase number of resistance providing cells. Now let us discuss cancer. Cancer is characterized by uncontrolled growth and division of certain body tissues. It is more common in people between 40 to 60 years of age. It is one of the chief killers of modern world, so as one of the most dreaded disease of human beings. According to World Health Organization report, about 10 million people throughout the world are diagnosed every year as having cancer. Cancers cause about 4 million deaths annually at the world level, whereas in India, more than 1 million people suffer from cancer and a large number of them die from it annually. Now let us discuss characteristics of cancer. Cancers show uncontrolled mitotic divisions of cells due to breakdown of regulatory mechanisms of growth and differentiation of cells. Cancer cells grow much more than normal cells. A tumor or neoplasm is formed. However, no tumor is formed in leukemia. These cells have less survival capacity or capability. Cancer cells are less adhesive than normal cells, so these generally move through the tissue to spread cancer to different parts of the body. These cells produce angiogenic factors which stimulate blood vessels to supply more nutrients for their growth. Cancer cells do not undergo differentiation. Normal cells show contact inhibition, which prevent uncontrolled growth in other cells, but cancer cells have lost this property. These cells have high invasiveness, that is, high tendency to spread to healthy cells. Now, etiology or study of cause of a disease. Cancer is neither contagious nor hereditary disease. The agents which cause cancer are called carcinogens. The carcinogens are of following types: physical agents, ionizing radiations like X-rays, gamma rays. They rupture DNA strands and induce mutations to cause cancer. Non-ionizing ultraviolet radiations may cause DNA changes leading to cancer. Physical irritants like certain foods, constant heat exposure may cause cancer. Then chemical agents like caffeine, polycyclic hydrocarbons, heavy metallic ions are carcinogenic. Sex hormones like testosterone, estrogens may cause prostate and breast cancer. Smoking, chewing of beetles may cause lip, mouth, and lung cancers. Dye workers using aniline dye have a risk of blood cancer. Then biological agents. Cervix cancer is caused by viruses. Tumor-causing viruses, Epstein-Barr virus, herpes simplex type 2 virus are cancer-causing viruses and named as oncoviruses. Now, types of cancers. Cancer types are studied under two headings. First is on basis of original tissue from where they arose. These are of following three types. Carcinomas. These are derived from epithelial tissue and glands. For example, cancers of lungs, skin, breast, stomach, pancreas, etc. Cancer of glands is called adenoma. That of melanocytes is called melanomas, and of muscles is called myoma. Now, sarcomas. These cancers are located in connective tissue and muscular tissue and derived from mesoderm. For example, cancers of bone, cartilages, tendons, adipose tissue, lymphoid tissue, etc. Cancer of bone is called osteoma. That of adipose tissue is called lipomas, and that of lymphoid tissue is called lymphoma. Then leukemias or blood cancer. It is characterized by abnormal increase in WBC count due to their increased formation in bone marrow. It is common in children below 15 years of age. Bone marrow transplantation is recommended in these cases. Then, on basis of their spread, tumors are of two types: benign or non-malignant tumors. These remain confined to their organ affected and do not spread to other parts of the body. These need immediate removal as it causes pain and may become malignant. Then metastatic or malignant tumors. These initially show slow growth, and this stage is called latent stage, but later on growth rate becomes very rapid, causing overcrowding and damage to normal cells. But later these enter metastasia stage, that is called stage of secondary growth, in which cancer extends to neighboring cells. Small pieces of primary tumor break off, carried by blood or lymph to other parts of body where these form secondary tumors. This process is called metastasis. The sites of metastasis are lymph nodes, lungs, long bones, liver, skin, and brain. Now, cancer detection and diagnosis. Early detection of cancers allow the disease to be treated successfully in many cases. Cancer detection is based on biopsy and histopathological studies of the tissue, blood and bone marrow tests for increased cell counts in case of leukemias are taken. In biopsy, a piece of suspected tissue cut into thin sections is stained and examined under microscope. This type of study is called histopathological study, as study of tissues take place. The techniques like radiography, use of X-rays, CT, that is computed tomography, and MRI, magnetic resonance imaging, are very successful to detect cancers of the internal organs. In computed tomography, we use X-rays to generate a three-dimensional image of the internals of an object. MRI uses strong magnetic fields and non-ionizing radiations to accurately detect pathological and physiological changes in the living tissue. Antibodies against cancer-specific antigens are also used for detection of certain cancers. Now, therapy. Cancer can be controlled in early stages. Radiation therapy. Cancerous part is exposed to ionizing radiations like X-rays, which destroys the cancerous cells. Then hormonal therapy. The hormone causing cancer is treated by another hormone which neutralizes carcinogenic hormone. For example, breast cancer is treated by testosterone. Then surgery. It involves the removal of entire cancerous tissue. Then chemotherapy. A number of chemicals like 6-mercaptopurine, 6-aminopterin kills the cancerous cells. These drugs have side effects like hair loss, anemia, etc. Then immunotherapy. It involves natural anti-cancer immunological defense mechanisms to be strengthened. Monoclonal antibodies, which are used in various ways, for example, radioimmunotherapy for treatment of cancer is used. With this, we come to close of today's lecture. I hope you must have understood it properly. Thanks for watching.